Can somebody confirm you can hear me?
I can hear you.
I can hear
Thank you.
you.
Sorry for being late, but there were meetings all day today at Clark, and I didn't realize how long it would take me to get home. I got in rush hour traffic, so it took me longer to get home than I anticipated. Are there any questions before we begin? All right, let me share my screen here. Let me remind you, you need to, oops, that's not my screen sharing. Let me go to Zoom. Let me remind you, you need to be working on your unknown project. You also be needing to be working on your infectious disease project. With the infectious disease project, you need to get it okayed by me before working on it.
I had a question.
Hmm.
I didn't see any uploads of the lab or lecture from Thursday last week, and I'm wondering if you did upload them and I missed them, or if those are still going to be uploaded.
If you put it in Thursday, I think I did it on Friday, although if you requested a test that took 2 days or more,
Oh, no,
then it would have waited until
the
Monday.
lecture video, the one you record for the Zoom.
Oh, you're saying I didn't upload the
I didn't see them, so
video.
I don't know if I
Ah,
just missed it or if it's
no, it could be I haven't uploaded that. I'll have to check on that. Okay.
okay. And
All
then
right.
I was
I don't
wondering
do
if
that
you've
now.
uploaded the slide PDF for chapter 6 yet.
I do not know about that. Let me go to YouTube and I
Thank
can
you.
answer at least the question of if I've uploaded lecture 6. Another thing I should mention is that a number of you are not viewing the lab, and you guys are going to be confused on especially the lab projects, but the lab in general. So please do view the lab, and I can check and find out who you are. I haven't done that yet. I'm not going to probably do that, but you do need to view the lab. So let me see. How do I upload? There we go. So it looks like the last thing I've uploaded was the lab on 5/8. So that means on 5/10, I have not uploaded those correctly. If I haven't uploaded it after a day, go ahead and send me an email and I will take care of it. Obviously, you can always mention it in class, although that would be several days later. Let me get the,
Okay,
oops, wait a
thank
minute, I
you.
gotta name it. I'm trying to upload the lecture file now. Now, it won't be uploaded for at least 15 minutes because um, we'll have to do a few things and then it has to put the, oh, what do you call it? The subtext in there. So there, I'm uploading it now and it'll be probably ready in about 15 minutes. Let me shut down the mail here. And I'll leave that there. All right. Any other questions? I did a number of grading over the weekend, so go take a look at your grade book. I'm not sharing my screen, am I? Yeah, I am. Let me stop sharing my screen. Um, I can tell you what I got graded if I go to the grade book, but I need to stop sharing my screen to do that. As of last night or this morning, I got the unknown project reports, not the reports, but the data sent out. I got quiz 1 and 2 graded. I'm not sure I graded 3C now. I don't think I've graded quiz 3C. So I'll try and work on that tonight. And I got the extra credit assignments due now from week 2 to week 5. The lecture worksheet for lecture 6 is graded. And I have the labs graded for both the first and the second time through lab 3. And then I got the lab 6 graded last weekend. I have not graded lab 7 yet. Looks like I've got 8 to do too. And I'll try and work on lab 7 tonight, so it'll be done by Wednesday. If I do not get lab 7 done by Wednesday, then the second grading will be done not this Saturday, but a week from this Saturday. Any question about any of that? All right, let's get going. Let me get out of the grade book. Then I'll share my screen again. All right, you should be seeing my screen. Today is August 12th. Come on, let's move. And that looks like I got the wrong one here. Yep, that's spring. So today is August 12th, and we are supposed to be starting chapter 8. I think we did get started with chapter 8. And let me remind you, you do have a quiz this Wednesday. The quiz will be over lab 7, chapter 5, and chapter 6. I see why you want the notes for chapter 6. Let me check on that. I don't have chapter 5 even up. All right, the lecture for chapter 5 and 6 are now gone up.
Thank you.
All right, let's go on to chapter 8. Genetics. I have that we had talked about DNA replication. That one strand is used as a template to make a new strand. And we're about to start RNA transcription. Any questions on anything so far? All right. Unlike DNA, where all of the DNA, unlike DNA replication, where all of the DNA is replicated, in transcription, only the genes are transcribed. The non-coding region of DNA is not transcribed. Now, for bacteria, most of the DNA is coding DNA. So most of the DNA actually codes for a gene. However, in eukaryotes, most of the DNA codes for non-coding DNA, meaning most of the DNA is not coding for a gene in eukaryotes. Any question about any of that? So RNA transcription is the synthesis of RNA from DNA. DNA is used as a template to make the RNA. DNA is transcribed to make messenger RNA, tRNA, and rRNA? I'm not sure I've ever asked this to you, but when the DNA polymerase is transcribing RNA, how does it know to make messenger RNA, tRNA, or rRNA? Can anyone answer that? It's a simple genetic question. Nobody's going to take a guess. All right. The RNA polymerase doesn't need to know which RNA to make because it's simply transcribing the DNA to make RNA. And if the gene codes for protein, it'll make messenger RNA. If the gene codes for tRNA, it will make tRNA. And if the gene codes for rRNA, it will make rRNA. So the RNA polymerase doesn't need to know what type of RNA to make. It'll simply transcribe the gene, and then that will determine which RNA gets made. Most of the genes code for messenger RNA because most of the genes code for a protein. But a few of the genes code for tRNA and a few code for rRNA. And it's a handful in one and like a dozen in the other. So, like I said, most of the genes clearly code for messenger RNA. And then if you want to amaze your friends, you tell them, yes, most of the genes code for messenger RNA, but most of the RNA in a cell is actually rRNA. That's just a neat fact to know. And that's because rRNA is needed to make the ribosomes. Obviously, there's R protein, meaning ribosomal protein that makes the ribosome as well. But there's a lot of RNA in the cell, which is rRNA. And that's the majority of the rRNA in a cell. Any question about any of that? All right. RNA transcription begins when the RNA polymerase binds to the promoter. A promoter is a region of the DNA upstream of the gene. And the promoter, all it codes for is RNA polymerase bind here. And then the RNA polymerase binds there and then moves down the DNA, transcribing the DNA as it's moving down it. Any question about that? Transcription proceeds in the 5 to 3' direction. So it only goes in the 5 to 3' direction. And that means that one strand of DNA will be transcribed in the 5 to 3' direction. Usually in eukaryote DNA, the genetic material is on one strand. It's only in viruses where both strands of DNA tend to have genes on it. And then the RNA polymerase will transcribe the DNA until the RNA polymerase reaches a terminator. A terminator is a little region at the end of the gene that actually codes for the RNA polymerase to fall off. When the RNA polymerase falls off, obviously the transcription will stop, and then the RNA will be fall off the RNA polymerase. And then the RNA can move into the cytoplasm if it's a eukaryote cell. It's already in the cytoplasm if it's a prokaryote cell. And then the RNA can engage in protein translation. Any question about any of that? All right, there's an actual electron microscopic picture of RNA transcription going on. That right there is a little bit of RNA being transcribed. This is the DNA.
How can they tell which Wait, which is RNA and which is DNA in the...
Well, I know this is the RNA because it's shorter than the
Okay.
DNA. I'm not sure.
I was just curious how they could tell the difference when they're looking through the
Well,
microscope
it's
that way.
oh gosh, what is that? Fluorescent microscopy in an electron microscope. So um, it could be that, well, in fact, both of them are lighting up. So um, so to answer your question, I don't really know, but
Oh,
I
I
know
was just
that
curious.
RNA because it's smaller than the DNA. And
Okay.
all right, so this is showing you the model of RNA transcription. Let's go ahead and go through our YouTube video. Come on. Having trouble getting it to load. Let's end this. Not sure what that is.
It's time to retrieve all those files you thought you
After a
lost.
commercial.
If you have data stored on a laptop or computer that no longer works,
Proteins are contained in our DNA. DNA contains genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a more distant location. Motor region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule.
Okay, let me just point out something here, and that is initially when the RNA is being made, it's going to bind to, hydrogen bind to the DNA. But you see, there's only the start of the RNA that actually does that. After that, the RNA falls off and it's not binding to the DNA, and that allows the DNA to snap back from the single-stranded to double-stranded. Only single-stranded DNA is transcribed. Only single-stranded DNA is also replicated. Any question about any of that?
No.
Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other. The strand of messenger RNA that is made during transcription includes regions called exons that code for a protein, and non-coding sections called introns.
All right, this is something that's only in eukaryotes. I'll talk about this when I'm finished with most of RNA transcription.
In order for the messenger RNA to be used in translation, the non-coding introns need to be removed, and modifications such as a 5' cap and a 3' poly A tail are added. This process is called intron splicing and is performed by a complex made up of proteins and RNA called a spliceosome. This complex removes the intron segments and joins the adjacent exons to produce a mature messenger RNA strand that can leave the nucleus through a nuclear pore and enter the cytoplasm to begin translation. How is the information in the mature messenger RNA strand translated into a protein? The nitrogenous bases are grouped into 3 letter codes called codons. The genetic code includes 64 codons. Most codons code for specific amino acids. There are 4 special codons, 1 that codes for start and 3 that code for stop. Translation begins with the messenger.
Okay, any questions
As
about
I've-
that?
No.
Okay, thank you. So transcription begins when the RNA polymerase binds to the promoter. And oops, it's saying my internet connection is unstable. If you're not hearing something, just get me to repeat it. Not sure why it's unstable, but it is. And when the RNA polymerase binds to the DNA, a helicase also binds to the DNA, and then the helicase moves down 5 to 3 prime, making the double-strand DNA single-stranded. And as I mentioned initially, the RNA, when it is transcribed, will be bound, hydrogen bound to the DNA. And right here to there, this messenger RNA is going to hydrogen bond to the DNA. But after this point, the RNA then falls off the DNA. The DNA is now single-stranded, and the DNA can snap back where it becomes double-stranded again. Any question about any of that? And then the RNA polymerase moves down the gene, transcribing the DNA as it moves down until the RNA polymerase reaches the terminator. The terminator codes for the RNA polymerase to fall off the DNA, and that will release the RNA from the RNA polymerase as well as from the DNA, although the DNA is only hydrogen bound to the RNA right here. It's only a little region. Any question about transcription? All right, let's talk about translation. So the RNA, if it's a messenger RNA and it's transcribed, it'll then move into the cytoplasm. In prokaryotes, the messenger RNA is already made in the cytoplasm, so it doesn't need to move into the cytoplasm. It's already in the cytoplasm. And the other RNAs come together to translate the protein from the messenger RNA. So messenger RNA codes for the protein, and how that coding is done is messenger RNA is read in 3 group of nucleotides that we call a codon. 3 nucleotides code for 1 amino acid. However, when the ribosome binds to the messenger RNA and then starts moving down the messenger RNA, initially, the messenger RNA is not translated into protein. The ribosome only begins translating the messenger RNA into protein once the ribosome reaches the starting codon, AUG. So the messenger RNA upstream of AUG is not translated into protein. Any question about any of that? And then the messenger RNA, AUG, codes for the first amino acid and tells the ribosome begin protein translation here. So AUG you should know because it does 2 things. It tells the ribosome begin protein translation here on the messenger RNA. And anything upstream of that will not be translated into protein. And then AUG also codes for the amino acid methionine. That means every protein, when it's first being made, will always have the amino acid methionine being the first amino acid. Well, here's a question for you. What happens if we have a protein that you don't want it to start with the amino acid methionine? What do you think a cell does? You want to have a guess? There's only a few things a cell could do. It could change the amino acid methionine to the correct amino acid. It could remove the methionine, or what else? Maybe something else that I can't think of now.
I'm so sorry. I didn't hear the question. Could you repeat it one more time?
All messenger RNAs only begin protein translation at AUG, which codes for the amino acid methionine. My question was, if the protein doesn't want to start with the amino acid methionine, what do you think the cell does? Does it change methionine to another amino acid? Does it remove methionine? Or something else?
Would it just remove it?
Yeah.
Like it would still start at the same part?
It will just remove the methionine, and then the next amino acid will be the amino acid starting the protein. So if it doesn't want to start with methionine, it just removes it. Because every protein, when it's first made, always will start with methionine, because that's what AUG codes for. Any question about any of that? And then somebody have a question? And then the ribosome moves down the messenger RNA, reading them 3 nucleotides at a time, or 1 codon at a time, because a codon is 3 nucleotides, and translating the RNA into protein. And that will happen until the ribosome reaches a stop codon. There's 3 of them, so you don't need to know them. But UAA, UAG, and UGA are the stop codons. And the stop codon tells the ribosome, stop protein translation here. And the stop codon does not code for an amino acid. Any question about any of that?
I had a question.
Uh-huh.
So when the tRNA sees the stop codon, nothing happens. It just stops, like, the chain, right?
No further amino acids will be added if the ribosome reaches the stop codon.
Okay.
All right, let's talk a little bit about the genetic code. Oops. Trying to move that out of the way. There we go. So translation reads the nucleotides 3 at a time, and that's a codon, which codes for 1 amino acid. Organisms have about 20 different amino acids. Humans have exactly 20 amino acids, and most bacteria also have about 20 different amino acids. The number 1, highest, is only 23, which is not much higher than 20. But most bacteria also only have 20 amino acids, but there are 64 possible codons. And why is that? If there's 64 codons, then there could be 64 different amino acids, but there's only 20 different amino acids. And what happens is there's more than 1 codon for the majority of the amino acids. There are a few, I think there's 2, where there's only 1 codon coding for that 1 amino acid. and that includes AUG coding for methionine. The other one you don't need to know, but UGG code for tryptophan. Those are the only two amino acids that only have one codon coding for that amino acid. All the other amino acids have multiple codons coding for the amino acid. Like phenylalanine has two codons coding for it, UUU and UUC. Serine, on the other hand, has four amino acids coding for it, four codons coding for one amino acid, serine. And then leucine actually has six codons coding for that one amino acid. So UUA, UUG, CUU, CUC, CUA, and CUG are all messenger RNAs that code for, well, I should say they're messenger RNA codons that code for one amino acid, which will be leucine.
Does the code affect it in any way, like affect its function or where it goes or what it does?
Are you talking about, does the codon CUU affect the making of the protein or anything about the protein differently
Yes.
than the codon CUC? Is that what you're talking about?
Yes.
Yeah. No, it doesn't. So both CUU and CUC code for the amino acid leucine. This is known as degeneracy of the code, where more than one codon can code for the same amino acid. All right, let's talk about translation of messenger RNA. Let's watch a little video first.
Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules, including mRNA, the small and large subunits of the ribosome, tRNA, and finally, the release factor. The process is broken into three stages: initiation, elongation, and termination. Let's see the process in action. Eukaryotic mRNA, the substrate for translation, has a unique 3' end called the poly A tail. mRNA also contains codons that will encode for specific amino acids. A methylated cap is found at the 5' end. Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the aminoacyl, or A site. The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid, the first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters. These are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and new tRNAs enter. When a stop codon is encountered in the A site, a release factor enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and the newly formed protein is released.
All right, any questions about that? So let's go through protein translation in a little more detail. So remember, we have DNA coding for the information. That information is translated into messenger RNA in RNA transcription. And then that message is translated into protein in translation. So here we have our messenger RNA, and it codes for the protein. The ribosomal subunits come together and bind to the messenger RNA. Normally there's more than 2 nucleotides before AUG. Normally it's a stretch of messenger RNA. I don't know how long, but let's say something like 20 to I don't know, 50, 100. And then the ribosome moves down the messenger RNA until it reaches the codon AUG. AUG codes for begin protein translation here and begin with the first amino acid methionine, meaning AUG plays 2 functions, telling the ribosome begin protein translation here, and then it also tells I guess the ribosome and the tRNA that the first amino acid should be methionine. Here's a question for you. If the ribosome moves down the messenger RNA, what's going to happen if it reads AUG, like right here? What's going to happen?
It's
You're going to skip it?
start.
What?
It will start, right? Or no, because there wasn't an N.
It's already started here. So yeah, AUG does code for start, but it's already started. So what
So
it's
it's
going to
gonna
do is
stop?
it's going to code for the amino acid methionine. Any questions about that?
So if it starts with AUG, can there be another AUG anywhere in that code?
Yeah, like I said, if ever the amino acid in the protein is supposed to be methionine, it's going to have AUG down here. Any questions about that?
No.
All right. And I'm making it a little bit simpler because sometimes you can have a protein which starts here at the first AUG, but other times you can have a protein starting at the second AUG, and you guys don't need to know that.
I had a kind of related question.
Uh-huh.
So, just to clarify, because I get them confused, is transcription DNA to mRNA and then translation mRNA to protein?
Correct.
Okay, thank you.
All right, so to get protein translation to happen, you need the messenger RNA, which has the code for the amino acids to put in. You need ribosomal RNA, and that is in the ribosome. There's also ribosomal protein in the ribosome, but you do need rRNA in the ribosome. You also need ribosomal protein in the ribosome. And then the third RNA is tRNA. It is actually the molecule that reads the codon and then inserts the correct base. So everyone says that the ribosome binds to the messenger RNA and then reads the codons, but actually it's the tRNA which is coming in and reading the codon and then bringing in the correct amino acid. So it's tRNA that's actually doing the reading. And how the tRNA does the reading is the tRNA has a short region called an anticodon, and it will hydrogen bond to the codon. So AUG will have the corresponding U, which is the complement of A, and then U will have A, so A is the complement, and then C is the complement of G. So the anticodon will actually hydrogen bond to the messenger RNA. Any questions about any of that? And then the tRNA brings in the correct amino acid. So UAC only has the amino acid methionine on it. And that is how the amino acid methionine gets added. Any question about any of that? So these are the components you need for protein translation: the messenger RNA, the tRNA, and the ribosome. The ribosome gets together the components and then moves down the messenger RNA until it reaches AUG. Nothing happens. Once AUG is reached, it tells the ribosome begin protein translation here, and then the first amino acid will be brought in by the first tRNA, which will have the anticodon UAC, and that will hydrogen bond to the messenger RNA. The ribosome has this channel in it, allowing the tRNA to come in, and the ribosome stabilizes this tRNA and this link here, because the hydrogen bonds between AUG and UAC is only going to be 2 here, 2 there, and 3 there. And as you know, this will be at room temperature at least, maybe 37 degrees Celsius. And hydrogen bonds are very weak. We mentioned that before, and they will break all the time. So what is there? 3. So there's only 7 hydrogen bonds holding it together. So you need the ribosome to stabilize it so that the tRNA can actually hydrogen bond to the messenger RNA. Any question about any of that? All right. The ribosome actually has 3 links in it. Only 2 are used. So only 2 are shown here. And then the second, we'll call it a channel in the ribosome, can allow the second tRNA to come in, and it will be the correct one, meaning this second tRNA will have the anticodon AAU, which will bind to the UA, bringing in the correct amino acid for the second codon, which in this case will be leucine. Any question about any of that? All right. Then the methionine is removed from the first tRNA, and that's happening by an enzyme in the ribosome. And then that methionine is chemically bound to leucine. Once again, an enzyme does that, and that enzyme is the enzyme of the ribosome. And if you remember, I mentioned this is the only enzyme we're going to talk about, which is actually an rRNA enzyme. It's part of the ribosome, and it actually removes the first amino acid from the first tRNA, and then takes that amino acid and binds it to the second amino acid on the second tRNA. Any question about any of that? Oh, you don't need to know the P site and the A site. You just need to know that the ribosome has two channels in it to allow the tRNA to come in and then hydrogen bind to the codon. And that's how this codon codes for the amino acid, the correct amino acid, meaning the tRNA has an anticodon, which binds to, in this case, AUG. And this codon only has or anticodon only has the amino acid methionine on it. Once the amino acid is removed from the first tRNA, that tRNA is then free to leave the ribosome. When that happens, when the tRNA moves out, the ribosome then moves down a codon. Let's go back. So before, the ribosome was actually at AUG and UUA. And now the AUG is outside the ribosome, and the second codon is now moved into the first codon, and the third codon can have the correct amino acid be brought in by a tRNA, which will hydrogen bond to the codon. Any question about that? And this is something you don't need to know, and I actually can't explain it. But, let's go back here. And that is this tRNA used to be in the second position of the ribosome. When the ribosome moves down, this tRNA then moves to the first position. So this channel must be connected, or guess I should say this channel must be connected to this channel, because this one moves from this position to that position. Any question about any of that? And then the third tRNA comes in, bringing the correct amino acid, which will be what GGU codes for. And then the enzyme will remove the leucine from this tRNA and then bind the growing amino acid chain. It's not really a polypeptide because there's only two of them at this point, but it'll bind the leucine and methionine to the third amino acid that was brought in, the glycine. And that's a covalent bond. And both the removal and of the amino acid and the attachment of it to the next amino acid happens by an enzyme, which is an rRNA enzyme, and it's in the ribosome. Any question about any of that?
So is the enzyme just called an rRNA enzyme, or is there like a specific name we need to remember?
There probably is a specific name for that enzyme. I don't know what the name is. All I know is it's part of the ribosome.
Okay.
Okay. And I'll have to even look up to see if they have a name for it, but I don't know. The only thing you need to know is it's an rRNA enzyme.
Okay, thank you.
All right. So when this tRNA is now released of its amino acids, it's free to then leave, and that will allow the ribosome to move down one further codon. And once again, the tRNA, which was in the second position, moves to the first position. Let's go back. AAU? I'm not sure. CCA. Oh, there it is. AAA. Here it is. It's coming in. So it used to be in the second position, but now it's moved to the first position. And this happens as the ribosome moves down the messenger RNA. It will read the codons, allowing the correct tRNA to come in, bringing the correct amino acid. Here we have an AUG, actually, bringing in another methionine right here. And that will happen as the ribosome moves down the messenger RNA. And that will continue to happen until the ribosome reaches a stop codon. The stop codon codes for the protein translation to stop, as well as it's coding for the ribosome to fall off the messenger RNA. And it'll disassociate, and that will also release the growing protein. So now that's the finished protein product. So the polypeptide, which is now the finished protein, is released, and the ribosome will fall apart into its subunits. The tRNA will come out as well. And then the protein, which initially was only in the first level of protein levels, meaning primary level, where it's simply the amino acid one after the other. But once the protein is released, the nearby amino acids can start interacting with each other and forming forces of attraction and forces of repulsion. Remember, when the growing protein is growing, it's not a protein yet, because you have to add the last amino acid to it before you have the finished protein product. Well, finished to the sense that we have synthesized it. Obviously, you can get the protein modified in the Golgi apparatus, and we're not going to talk about that here. Any question about any of this? All right. So let's talk a little bit about complications. Here we have the gene, and as you can see, we have 1, 2, 3, 4, 5, 6, 7, 8 messenger RNAs being transcribed from this one gene. And this we see all the time in cells where the gene will transcribe more than one messenger RNAs from that same gene. Any questions about any of that? And it started here because this is the longer messenger RNA. As you come down, we have shorter messenger RNAs. So this one looks like it's maybe only 2 So this one's just shortly been started. And the RNA polymerase will move down the gene, making the RNA longer. And then we have another complication in bacteria, and that is the messenger RNA is being transcribed in the cytoplasm. The ribosomes can actually bind to the messenger RNA and then start protein translation before the messenger RNA is finished being transcribed. Meaning this one here, the messenger RNA is very short and it needs to be made at least this long, probably longer. And yet we have 2 ribosomes bound to that short messenger RNA, beginning to translate the messenger RNA into protein. And this one has a large number of ribosomes bound to it. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22. About 22 ribosomes bound to that messenger RNA. Each of them will begin translating the messenger RNA to the into the protein. The protein isn't shown here, or if it is, it's very grainy, and I can't see it there. But it doesn't look like the protein showing here. It looks like only the DNA right there. The messenger RNA is the string going through here, and then the ribosomes are bound to the messenger RNA. And there's a couple of complications. First, in bacteria, you can have protein translation beginning before RNA transcription ends. Like right there, protein translation is beginning before RNA transcription has gotten very far. The RNA has to be at least this long. And we've only got this long. Any question about that? This is only seen in bacteria and in other prokaryotes because in eukaryotes, the RNA is transcribed in the nucleus, and then the RNA moves into the cytoplasm where protein translation occurs. So in eukaryotes, you don't have this complication, but in prokaryotes, you do. And then the other complication is that the translation can begin before transcription ends, and you have multiple transcripts being made from the same gene. That last part you can get in a eukaryote. You can get multiple transcripts from the same gene in an RNA, but you would never get protein translation beginning before RNA transcription ends in a eukaryote, because transcription happens in the nucleus and translation happens in the cytoplasm. Any question about any of that? All right. Eukaryotes are a little easier, but they have their own complication. And we've already talked about it, and that is in eukaryotes, in a gene, you have regions of the gene coding for the protein, and these are called exons. And inside a gene, which actually is in the first messenger RNA being transcribed from this gene, you'll have intron regions of the messenger RNA, which do not code for the protein. Before the RNA can leave the nucleus and enter the cytoplasm, the introns have to be removed from the exons, and the exons need to be spliced together. The term for that, both removing DNA and then adding two sections of DNA together is called splicing. Only the spliced messenger RNA can leave the pores of the nuclear membrane, meaning there's something on the pore of the nuclear membrane that prevents the RNA transcript from going into the cytoplasm. It will not allow it to happen until the introns are removed from the messenger RNA. All right, any question about any of that? So that's the structure and function of genetic material. That was the flow of genetic information, DNA replication, RNA transcription, and protein translation. We've now covered all of these topics. Let's move on to regulation of bacterial gene expression. Gene expression is said to happen by the operon model of gene expression. And then after we're done talking about operons and gene expression, we'll talk about mutation a little bit. Then we'll talk about genetic transfer and recombination. And lastly, we'll talk about genes and evolution. I think that last is only one slide, maybe 2 slides. Any question about any of this? How are we doing time-wise? Oh, we have time. So regulation of bacterial gene expression. One way to regulate the contents of a cell is to control gene expression. There are some enzymes which are expressed at a fixed rate, so we're always making these enzymes a protein. And these are called constitutive genes, meaning the genes are always turned on. They're also called housekeeping genes because the gene is always turned on in the cell. And then there are other genes which are sometimes turned off. So all genes are expressed, meaning turned on at the same time. Well, maybe not all at the same time, but when you start putting cells into a new media, the genes will be expressed, and it will eventually happen to all cells where the genes will be expressed. Sometimes the genes are expressed at a fixed rate, and other times the genes are only expressed when they are needed. That's a very good way to control gene expression because you only want to make the product if you need the product. If you make the product and you don't need it, you're just wasting energy. And plants don't want to waste energy as well as photosynthetic bacteria or any microbe. So they turn on and off genes when they are not needed. Not all genes, because obviously some genes are expressed at a fixed rate, but many genes are not. And when they're not needed, the gene is frequently turned off. We will briefly mention that there are, well, about 4 or 5 ways to control gene expression. The first way is with an enzyme causing repression or an inducible enzyme, meaning causing induction. And this happens whenever the gene can change its gene expression. And if many copies of the gene are needed, then many copies of the gene will be used to express the enzyme. We have 2 ways of controlling whether the genes are on or off. It's repression and inducible. So some genes are repressible, meaning you have a protein which represses the gene expression of the gene of interest. And there are other systems which are inducible, where the gene is turned on by certain, what's the word I'm trying to say? The gene is turned on by certain factors of the cell, and when the gene is needed, you will begin transcription and translation of the gene. If you need a whole bunch of gene, you'll be transcribing more than one gene. We'll talk a little bit more about the difference between repressible and inducible systems, but for now, all you need to know is these are systems where an individual gets very little sun. Obviously, you got to walk from the car to the door of the store. And they used to be at Clark have multiple rooms where you would have everyone sit like in a circle or something. If the students aren't cooperating or not giving any answers, we'd say that is an inducible system. But once you start inducing the students by offering extra credit, you'll find they'll start talking. Any question about any of that? All right. There are other ways to control gene expression besides repression and inducible enzymes. You can control gene expression by pre-transcriptional control, which in most animals, when you have pre-transcriptional control, the maternal gene is the one expressed. And what's happening is the maternal gene is more likely to survive the harsh environment of the microorganism. And so people this weekend sent me a whole bunch of Lab 7, perhaps because they initially couldn't figure out how to nail it in place. Any question about any of that? All right, so we will talk about inducible operons. We will just talk about repressible operons in passing, so not much. We're not going to talk about repressible operons for epigenetic control, so you don't need to know that. I just want to introduce it to you so in your next class you'll be somewhat familiar with the regulation of gene expression using the Appian model. Okay, any questions? So if the gene isn't needed by the cell can turn the gene off. And an easy way to do that is just regulate the gene and turn the gene off. Any questions about any of that? All right, so we have the operon model to explain gene expression. A bacterial operon is a supergene containing one or more structural genes of interest. So when we're talking about the blue-eyed human gene, we're talking about a structural gene. When we're talking about the gene coding for the enzyme lactase, we're talking about the structural gene Z. Y is the gene coding for the protein to bring the sugar lactose across the cell membrane, so it's the transport protein. And then A is a gene that is involved in the metabolism of the sugar lactose. Once you split lactose into, I think it's glucose and beta galactose, the A gene is important in helping change beta galactose to the sugar glucose. The point is, in an operon, you can have one or more structural genes, at least in bacteria. And then the operon includes the structural genes as well as the controlling regions around the structural gene. One controlling region we've already talked about is the promoter. The promoter is the region of DNA upstream of the structural gene that codes for the RNA polymerase to bind to the DNA. We also have the operator, and then not shown here is the terminator, which follows the structural genes or gene. Any question about that? So we're going to talk about the lac operon, and we're only going to talk about one operon, the lac operon, which is an operon in bacteria having 3 structural genes in it, a promoter region, an operator, and then the terminator, which you probably put that in the end of the structural gene there. Any questions about that? You'll note that we have a regulatory gene, labeled the I gene here, that is not part of the lac operon. The regulatory gene has its own operon, and this regulatory gene makes a product that controls the expression of the lac operon. Now, normally, the regulatory gene controlling the expression of the operon is nowhere near the operon that it's controlling. Like in humans, most of the regulatory genes are on a different chromosome than the gene that the regulatory gene is controlling. In E. coli, most of the regulatory genes are just someplace else on the DNA. There's only one DNA molecule in E. coli, at least most of the time. And the regulatory gene is nowhere near the DNA that it's controlling. However, in the case of the lac operon, the regulatory gene is right next door to the operon that it is controlling. Any questions about any of that? All right, let me state a little bit of the difference between a eukaryote operon and a bacterial operon. A bacterial operon can have one or more structural genes in the operon. In a eukaryote operon, there is only one structural gene in each operon. How many of you drink milk or eat dairy products? Anyone going to admit it?
Nope, I can't.
You can't?
Yes.
Okay. All right, there's somebody who
No.
can. All right, so if you eat or drink milk or some other dairy product, do you have the enzyme for lactase, the enzyme to break down the sugar lactose? That's the
Yes.
Z structural gene.
Yes.
Yes. Do you have the gene that brings the sugar lactose across the cell membrane?
Yes.
Yes.
Yeah, you've got the Y structural gene. And you also have the A structural gene. So in eukaryotes, we have the same structural genes. It's just that instead of all of these structural genes being in one operon. In a eukaryote, we have 3 operons to control the metabolism of the sugar lactose. Any question about that? So in bacteria, you can have more than one structural gene in the operon. You always have one structural gene, but you could have more. And eukaryotes only have one structural gene in each operon. Let me see. The structural genes that are in the operon tend to be related genes. In the case of the lac operon, all of the structural genes are involved in the metabolism of the sugar lactose in one way or another. The Z gene, of course, breaks down the sugar lactose into beta galactose and glucose. The Y gene brings the sugar lactose into the cell across the cell membrane. And then the A gene is involved in converting beta galactose to the sugar glucose. All right. We're doing fine on time-wise. So regulation of a gene can be either induction or repression. Induction is where the gene is normally turned off and you have to induce it to turn the gene on. In the case of the lac operon, it is an inducible operon, meaning in E. coli, the lac operon is normally turned off. And the structural genes are normally turned off in E. coli. Why would E. coli normally have the lac operon turned off? How often would E. coli see the sugar lactose? Okay, let me ask it this way. Where do you find the sugar lactose?
In milk?
Dairy.
In milk and dairy products. If you have E. coli in the soil or on an apple, how often is that E. coli going to be around the sugar lactose?
At
Not
a
very
farm?
often, huh?
At a farm?
Yes.
Yeah, at a dairy farm, the E. coli might be around the sugar lactose. And obviously, if you have E. coli in your gut and you're eating milk products or dairy products, you would see the E. coli would see the sugar lactose. But if the E. coli is in the dirt or it's in on an apple, this E. coli is not likely to see the sugar lactose because lactose is really only found in dairy products. And so normally, E. coli will shut down the lac operon because E. coli is not going to see the sugar lactose. If it's not going to see the sugar lactose, why would you make these enzymes which aren't going to be used? And so normally in E. coli, the lac operon is turned off. How it is turned off is by this regulatory gene. It's not a part of the lac operon. In fact, it's its own operon, another operon. And it gets transcribed, making a messenger RNA, which is translated into a repressor protein. The repressor protein binds to the operator of the lac operon. When the repressor protein is bound to the operator, now when the RNA polymerase binds to the promoter, it starts moving down and then it bumps into the repressor protein and it cannot move any further. So the repressor protein stops the RNA polymerase from moving down the DNA. And then the structural gene, like the lactase enzyme, will never be transcribed. And if it's never transcribed, the gene is turned off and the protein will never be translated. Any question about any of that? So that's normally what happens in E. coli. The repressor gene gets made, making the repressor protein, which binds to the operator, and that stops the RNA polymerase from moving down the DNA. Well, here's a question for you. That RNA polymerase, is it going to be stuck there forever until that
No.
repressor protein falls off? The
Yes.
answer no is correct. And the RNA polymerase binds to the DNA in a stoichiomic reaction, and every once in a while, it will fall off. And that's good because you don't want the RNA polymerase to be permanently stuck in this position because it can't do its job in the cell. And so every once in a while, it'll fall off, and then it'll bind to another promoter. And of course, if it binds to the lac operon promoter, it will once again get stuck by the repressor protein. Okay, well, we need some way of turning on the lac operon, and the easy way to state it is that the repressor protein also binds to the operator in a stoichiomic reaction. So every once in a while, the repressor protein will fall off. And once it falls off, the polymerase can then transcribe the gene. If there's other repressor protein around, it will bind to the operator, and that will turn off the gene. I mean, this polymerase may get through, depending on how quickly the repressor protein binds, but only one messenger RNA will be transcribed, and the next polymerase binding will get blocked by the repressor protein. Any question about any of that? All right. Well, we need to have a way of turning on the lac operator, and how that happens is you have an inducer, which is actually allolactose. Allolactose is an isomer of lactose. One is right-handed and one is left-handed. I think allolactose is the right-handed, not that that's terribly important, and lactose is the left-handed. So whenever you have lactose, the sugar lactose around, you have allolactose around because they're just isomers of each other. And allolactose will bind to the repressor protein, inactivating it. When it's inactive, it cannot bind to the operator. And then when the RNA polymerase binds to promoter, it will transcribe the gene. Any question about any of that? All right, let's go ahead and watch the video on the lac operon.
The E. coli lac operon is an example of an inducible set of genes. These genes are responsible for the breakdown of lactose into sugars used for cellular metabolism. This inducible system also involves bacterial DNA, a repressor, mRNA, and the sugar molecule lactose. This animation will only focus on 2 of the 3 proteins encoded by the lac operon: beta-galactosidase and permease.
Whoops.
Gene expression can...
Let's go back here. I thought I stopped it, but I didn't. Beta-galactosidase is another name for the enzyme lactase. Permease is the name for the active transport protein that pumps the sugar lactose across the cell membrane.
Gene expression can be induced or turned on when a specific inducer molecule appears in a cell. For inducible systems, a repressor molecule prevents gene expression by binding to the upstream controlling region. Lactose is the lac-operon inducer molecule. After first appearing in the cellular environment, lactose passively enters the E. coli cell and binds to the repressor molecule. This binding releases the repressor from the controlling region. At this point, RNA polymerase can begin transcription of the operon. Here we show 2 of the 3 lac-operon genes being transcribed into mRNA. Ribosomes then bind to the mRNA, and the 2 proteins are translated. The first protein is beta-galactosidase, which breaks down lactose into 2 simple sugars. The second protein is permease, a membrane-bound protein. When embedded in the cell membrane, permease functions to provide a direct route for the lactose outside the cell to be imported into the cell. This import occurs at a much greater rate than the passive transfer we first observed. Because translation continues inside the cell, other permease proteins become embedded in the membrane. This further increases the rate at which lactose enters the cell. Beta-galactosidase breaks the cellular lactose into the simple sugars glucose and galactose. Once its concentration is greatly reduced, the lactose bound to the repressor are released. At this point, the repressor again binds to the controlling region and gene expression is halted. For all inducible systems like the lac-operon, it is the interaction of the repressor and inducer molecules that mediate gene expression.
Any question about any of that? Well, here's a question for you. This messenger RNA codes for how many proteins? One, two, three. Have we ever seen a messenger RNA do that before?
No?
Yeah. So in the lac-operon, one messenger RNA is made, transcribed, and then that messenger RNA would be translated into 3 different proteins. And we've never seen that before. Any question about any of that? All right. Any question about the lac-operon or operons in general? So if not, let's switch to a little bit different topic. We need to talk a little bit about mutation. A mutation is a change in the DNA. Mutations may be neutral, beneficial, or harmful. Here we're actually looking at a mutation. A mutagen is an agent that causes mutations. So like benzene is a mutagen, cigarette smoke is a mutagen, and they cause a change in the DNA. But you can also have a spontaneous change in the DNA, and this is called the spontaneous mutation. Spontaneous mutations occur in the absence of a mutagen. Any question about any of that? One mutation that I'm going to talk about a little bit is the mutation in the human population about 10,000 years ago, where in Northern Europe, everyone who had brown eyes, and the humans only had brownish eyes back then, meaning different shades of brown, there was a mutation in Northern Europe that gave rise to the bluish-eyed allele. And that mutation went on to become the the major allele, meaning the allele, most common allele in Northern Europe is the bluish-eyed allele. So they think 10,000 years ago, the bluish-eyed allele either had a selective advantage and it increased in population, or it was next to a gene that gave the blue-eyed allele a selection by the other gene was selected for. The point is, 10,000 years ago, humans initially only had one brownish-eyed allele, and then 10,000 years ago, that number increased to two, where we had bluish-eyed individuals and brownish-eyed individuals. And you can separate all humans into blue-eyed or brownish-eyed. Brown would be almost dark, like close to black, dark brown, light brown, and then greenish brown, which I think is called hazel. Green eyes, very sky blue, and then very light blue are all under bluish eyes. The question is, will there be another mutation in the human population, like causing yellow eyes? Dogs have blue eyes, brown eyes, and yellowish eyes, so it's possible in the future that humans may get the eye color yellow. Any question about any of that? It hasn't happened yet, but in the future it might happen. There are different types of mutations. There's a base substitution, also called a point mutation. This is where one base is changed in the DNA. There's three types of base substitution: a silent mutation, a missense mutation, and a nonsense mutation, and I'll talk about them in a little bit. There's also a frame shift mutation where one base is either added or one base is removed. It's called a frame shift mutation because when you add one base, you probably change the reading of the codon where that adding of the base is going to happen, because you're changing the codon, which could change the amino acid. But then every codon afterwards is changed because we've added a base, and only the first three. So let's say we have two bases. Well, actually, we need three bases, which code for a codon, and then we add a base between the second and the third. And now we have three bases, the third being a new one, which could code for a different amino acid. And then that next nucleotide, which used to be in the third position, is now in the fourth position, will be the start of the next codon. So all the codons after the addition or after the subtraction will be changed. Is that clear to everyone? And a frame shift mutation could be a multiple addition or a subtraction of a nucleotide. If you get two added or two subtracted, once again, you have the frame shift mutation, and then all the other frames of the codons could be changed. So all the other codons could be changed. However, if you have an addition of three or a subtraction of three bases, it's not a frame shift mutation, because let's say you pull out three bases and we had, I don't know, AUG and then AAA and then GGG, whatever the AAA coded for will be missing in the frame shift mutation. And it's not a frame shift mutation, so that one amino acid is missing. But then the second amino acid will be the, what would I say, GGG? And so one amino acid is changed, but all the others are the same. So a frame shift mutation is when you have an addition or a subtraction or a multiple addition or a multiple subtraction, unless it is a multiplier of 3. 3, 6, or 9 addition will not be a frame shift mutation because the codons won't have their frames changed. Any question about that? You might add an extra amino acid if you put in 3 bases, but then the codon reading will be correct. It's just that the protein will have an extra amino acid. Any question about frame shift mutation? All right, a macromutation was where an entire region of a chromosome is either added or deleted. A macromutation is not discussed in your book, so you have to get it from the lecture. And if you go online to look at a macromutation, you'll find that there are 2 different macromutations. One being where an entire region of the chromosome is added or deleted, and that's a mutation. The other is where you change a gene that controls a lot of genes. What the heck is that called? A controlling gene. It controls the expression of other genes. And that is not a macromutation in the type that I'm talking about. So learn the term as I'm giving it here, where a macromutation is where an entire region of a chromosome is either added or deleted. What is a macromutation? Where an entire chromosome is added. What do we
Like,
call that? Go on.
would like, sorry, my words just left me. Would, Down syndrome be like a macromutation?
Yeah, Down syndrome was the example I was trying to get at. It's a macromutation where an entire chromosome is added.
And then like, is it Williams syndrome, the one where one is like they're missing chromosomes?
I don't know. I'm not familiar with Williams syndrome. Do you want me
Okay.
to look that up or do you
No,
not?
that's
No.
okay.
All right. Why are we here? Oop. All right, let's talk about the base substitutions and point mutation. It can be silent mutation, a missense mutation, or a nonsense mutation. A base substitution where we had, what did we have here? ACA gets switched to ATA. Is that right? yeah, that's right. And it used to be UGC, coding for the amino acid cysteine. When you have the base substitution for G being switched to A, it codes for the amino acid tyrosine. This is a base substitution where one base gets changed. Any question about any of that? There are 3 types of base substitution. There's a silent mutation, sometimes called a neutral mutation. But I do want to state that when I stated mutations can be neutral, beneficial, or harmful, a neutral mutation is one where the individual has the mutation, gets no benefit out of it, but gets no harm out of it. A beneficial mutation is one where the individual having the mutation benefits from it. Let me give you an example of it. There was somebody who had a fruit fly lab. And here, this is a good example. He had his buckets. Can you guys see this? I can't see it, so let me end the show here and stop the share. He had a fruit fly lab where he had the container and you just put a cotton swab or paper towel in the bucket, the container, to keep the flies from flying out. And he was very lazy and he didn't change out the dishes, and the fruit flies would come out and the larvae would crawl to the side and then they would pupate on the side of the glass. He was so lazy that there were so many pupa on the side of the glass that the larvae coming up couldn't find a region of the glass to become a pupa. And so what happened was there was a mutation in his lab where some of them actually crawled up onto this is a paper towel here, on the lid, and then they started pupating inside the bottle on the paper towel or the cotton swab. And those were beneficial because there was no room on the side, so that mutation increased in number in his lab. Any question about any of that? A harmful mutation is one where the individual is selected against, and an example of that is sickle cell anemia. An individual with 2 sickle cell genes is unlikely to live very long into adulthood. Usually they die in the early 20s. Obviously, modern medication can make them live longer, but they're cell sickle, and then they have a whole bunch of other health problems. Any question about any of that? And that's the homozygote, meaning a person with 2 of the sickle cell genes are selected against. I don't want to talk about the heterozygote. All right, let me share my screen here.
It's not super important, but I was actually born with a base substitution mutation that's dominant.
And do you know the mutation?
Um, I have classical Ehlers-Danlos syndrome, so my collagen is affected. So the substitution is, I believe, glycine for arginine.
Okay.
I'm not
So
probably
that's
not
a
saying
base
those are right.
substitution. And also, let me state what the silent mutation is. Your mutation and where you change the amino acid by changing the DNA and change the amino acid, that's a missense mutation. A change in the one base of DNA that results in a change in the amino acid. There is a silent mutation where you change the DNA, but it does not change the amino acid. For example, if you had UUU and it's mutated to UUC, the DNA got mutated, but the protein was not mutated because UUC still codes for the amino acid phenylalanine. And this is a silent mutation where the DNA is changed, but the protein is not. Any question about any of that? All right.
No, but I think we're out of time.
I know. Let me just talk about the last mutation and then we will end it there. And that is the nonsense mutation. That's where you change one nucleotide in DNA and it results in a stop codon. When you have a stop codon, you have a truncated protein because instead of having your normal protein, you only have a shortened truncated protein stopping at the stop codon. Anyways, that's a nonsense mutation. Any questions about mutations? Actually, it's 6:50 now, and this is a good place to stop. All right, if there's no questions, I will see you at 7 o'clock for the lab. Assuming we have a lab today. Let me take a look at that. Yeah, we have lab 9 today. All right. Bye.